Aloha, I'm Dr. Gail Grabowsky, and thank you so much for joining me for this talk today. I knew that I had to give it when I heard that the theme of the Hawaii Conservation Conference was managing for abundance, because this talk is all about the abundance of reef fish that are collected for the aquarium trade. So, the title of this talk is Abundances of Aquarium-Collected Coral Reef Fishes in Protected and Open Access Areas of Oahu, Hawaii. So there are no published studies from Oahu of the effects of the aquarium collection industry, trade, or trafficking, depending upon who you talk to, whether we have impacted abundance from Oahu. There are many studies from the Big Island, from West Hawaii. So you are probably hearing for the first time about the effects of aquarium collection on Oahu. This study was conducted by myself and Dr. Daniel Thornhill, who is the Biological Oceanography Program Director at Auburn University, and I'm the Environmental Programs Director and the Interim Dean of Natural Sciences and Mathematics for Chaminade, where I've been for 23 years. And the impetus for this study, the reason that we conducted it, there's a couple reasons. One is that my sister is actually a fish biologist and was rearing soft corals, and she knew a lot about the aquarium industry. She lived in Florida. And so as she talked to me, I thought, wow, I wonder how we're doing on Oahu. I wonder if there's an impact. And then also learning about some of the studies as I started to read the literature, the methodology is really not super complex, and I thought students could do this. And so I was co-PI on a grant that Dr. Mike Hadford was the PI for, the UMEB, and then a URM grant that involved Pacific Island students doing research internships. So this seemed like a good summer project. So the students are actually the data gatherers of this study that I'm reporting on today. So we decided to model the study after Tissot and Hallaker, 2003, and they assessed the effects of aquarium collecting in Kona. And so they did it because there really had been no previous studies that conclusively documented the magnitude of the effect of aquarium collecting on natural populations of reef fish, even though the issue had become contentious between stakeholders in many places. So I should say many places in Hawaii, and particularly in West Hawaii, but also on Oahu. And so it was time to gather some data, make some form some database beliefs. And so their methods were, they trained undergraduates just like I did, and they completed coral reef monitoring course, and they were trained how to ID fish, and they were also trained in the particular methodology, which I did with my students. And then the data that was collected, we did exactly the same thing. We compared differences in fish abundance along visual belt transects between collection sites where collecting was known to occur and control sites where collecting was prohibited. On the Big Island, they used the fishery replenishment areas and the other areas that have been protected longer. On Oahu, we have very little fully no-take zones, and very few, less than 1%, as most people know. And those are the marine life conservation districts. Hanauma Bay is one of them, as is Pupukea and Coconut Island. And so we used all three of those control sites, but because we heard of poaching and saw some poaching, we were left basically with just Hanauma Bay. So we used Hanauma Bay as our uncollected site that we call the protected site. And the sites that are where collection occur, we call those unprotected or open. So what Tissot and Hallaker found was that 7 of the 10 fishes targeted by the aquarium trade were actually significantly lower in abundance in areas that were subjected to collection versus their control areas. Their results suggested to them that the collectors had a significant effect on the population sizes. And they also were concerned that there was evidence that suggested the system of catch reporting underreported. So the data gatherers the Pacific Island students, I had 6. This study was conducted from 2009 to 2011 in the summer months. And the students were Paulino Balagat from Pohnpei, Winder Loiak, who is here from Majuro, Anthony Lecoq-Ficne here, who's from Yap, Nelson Nagrurikul, who was from Palau, Eva Sala from American Samoa, and Nicole Yamase from a number of islands in Micronesia. And they were wonderful field biologists in the water. All of them, you know, had experience growing up in the water. And these 2 guys were amazing. They were both wonderful fishermen. So the fish that we studied 16 species. I've color coded them here as Tissot and Haliker categorized their fish species. The collected species by their study are in yellow, and the non-aquarium or uncollected species are in green. Now, I'm going to tell you later that we could not trust that for our study because we worked 7 to 9 years later, and some of these fish in green here we found for sale. So we couldn't consider them uncollected. So we were left with the best comparison control being Hanauma Bay, the protected site that was well protected, and all the other open sites. So where did we count fish? We started out with a whole bunch of sites. Here's a map from one of the years. Not all of these sites made it in to this paper because there was some, I thought, very legitimate criticism that, you know, some of the habitat types were not the same. So what I did was to look at the NOAA, National Center for Coastal Monitoring and Assessment maps, and they have both a bathymetric geomorphological component and a biological component. And we only compared the fish in Hanauma Bay to the places we had counted that were the same by those measures. And so you do see some clusters here around and a point, one site here and a cluster here, and we did get some counts in over here. And if we look at the data from those maps, here is the geomorphological and the biological, and if you compare, you'll see that our Hanauma Bay sites both are like all of these sites. So we threw out a bunch of the sites and ended up with 17 sites. Our results found that 6 species were significantly more abundant in Hanauma Bay at a p.05 level with a t-test. So the 6 species that we found to be more abundant in yellow here with the asterisks are species I'm going to show you next in a table. So the ones that we found that were more abundant in Hanauma Bay are here with the aqua blocks, and of these 6 species, 4 of them are also the most collected aquarium fish on Oahu. I FOIA'd the state's collection data. It is here. Here are the grand totals for these years, and the #1 most collected fish, 92,000, yellow tang, is also a fish we found a significant difference in Hanauma Bay and everywhere else. Also, #2, the Kole tang, we found significantly more fish inside Hanauma Bay than outside. The 3rd most collected fish, Nasolidoratus, the orange spine unicorn fish, and also the Moorish idol. We did not find a difference in Forsythiger, and that's because they are super rare everywhere. And finally, with the fish that Tissot and Haliker didn't study, the raccoon butterfly fish, we added it because we saw them for sale, and so on the internet. And so we found a significant difference between Hanauma Bay. Also, the convict tang, which, as many of you know, is a food fish. Now, a few of these fish are also food fish, but 3 are not. The yellow tang, the Moorish idol, and the raccoon butterfly fish are never taken as food fish. So the difference, given that the habitats are the same and that collection does not occur in Hanauma Bay, is most likely due to collection. So our conclusions. We conclude that our results suggest that collection-driven population declines are occurring. We also conclude that, let's see here, 4 of the species based on the state's data are the most collected on Oahu. We also conclude that 3 of these species are not taken as food fish. So we can't say that the differences in abundance are due to fishing, food fishing. Finally, our results run parallel and congruent with the findings of
many studies, one of them, which I've quoted here, Williams and Walsh, which found that in Honolulu on the Big Island, of the 20 most collected aquarium species, 18 declined in abundance, with the species facing the heaviest fishing pressure, typically showing the greatest declines. It's exactly what we found. Our t-test revealed the highest significance in the species that are the most collected. So, to speak to some challenges and limitations in the study. One, there is no good baseline data for Oahu, so we can't say go back and look in the records. I've done that. I've looked at all the oldest species. I've looked at the great studies of Brock, but the methods for counting fish are so different that we cannot compare them, and I thought it was more important to use the methods on the Big Island. Also, the fish that are collected do vary over time. So we have a bit of a moving target here. As some fish become rare, other fish start to appear in the pet stores. Also, there was poaching on some protected sites, so that's why we only had Hanauma Bay. Also, let's see here, even Hanauma Bay is not a perfectly protected site. There's plenty of sunscreen in there, as we know, and a lot of people. So it's not pristine. Nevertheless, our statistically significant results, which are consistent with the collection intensity data and analogous studies from other islands, do make a convincing case for collection-driven declines in reef fish abundance. Finally, implications. In these days of nutrient pollution and climate change and overfishing, this is possibly another stress we're putting on reefs. And to make an ethical statement, is it right that we decide that fish sold for money is more valuable than their value on the reef when the reefs are so challenged? And finally here, many of the fish that are collected are herbivorous, and we know that herbivorous fish create turf algae, which is much better than macroalgae, and help to not allow phase shifts on coral reefs. Thank you very much for this talk, and I hope I have, for attending this talk, and I hope I have time for questions. And I'd like to thank all of these folks and my students. And with that, I'd love to take a few questions. Thank you.